This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
In the interest of time, I'm going to go ahead and introduce Susan. Hopefully people will continue to filter in. Thank you all for coming today to our joint Brain and Mental Health Hub and Cognitive Neuroscience Symposium. Professor Susan Perlman is here visiting us from Washington University in St. Louis. And she's a colleague of mine that I've known for a little while now because we share interests in development and neurobiology. So we were speaking about some potential collaborations and things like that, and I invited her to come to Melbourne for a bit. So she's here to speak with us about some of her work. I'm just going to give a brief overview of her bio. So Susan has a very multimodal research program. She focuses on developmental psychology and cognitive neuroscience. Her work uses biological measurements that predict school-age children who are most at risk for later psychopathology. So this is obviously of interest to many of us in this room. She uses fMRI, fNIRS, and psychophysiology measures, as well as eye tracking, behavioral, and questionnaire measures about cognitive and neural analyses and mechanisms underlying the development of emotion and its effects on social interaction. She's also done a lot of work with parent-child relationships, moment-to-moment interactive behavior, and other environmental influences on the family. Today, she's going to give us a broad overview of work she's been doing in our lab. I hope it inspires some creativity among many of us here about the types of questions we might be asking and methods we might use. So please join me in welcoming Susan.
Thank you, everybody. I am just so excited to be here. I've been loving Melbourne so far, and I'm really excited to hopefully collaborate with you guys and meet more of you. So, in the interest of time, I've made this kind of a overview of everything I've done in my career. So there's a lot here, and in the interest of time, since we're running late, I'm gonna skip the MRI slides. I do MRI and near-infrared spectroscopy, but people seem to usually want to hear more about the near stuff. So I'm gonna skip the MRI stuff so we have more time for questions. Okay, so my lab is called the Laboratory for Child Brain Development at Washington University in St. Louis, which you may have not heard of. It's a private university in the center of the country. What we do is focus on using multimodal imaging combined with behavioral and psychophysiological measures to understand child neural development and the influences upon it as a predictor of psychopathology onset. Okay, so I mostly study children in the preschool period. So that would be, let's call that broadly age 3 to 6. And why do I study this period? Well, it's a period, for those of you developmentalists, it's a period of intensive emotion regulation and executive function growth. You see that steep slope of both of those in that period. It's also a period of increasing independence. It's a period where children are moving away from being with maybe a primary caregiver at home to navigating social relationships, teacher relationships, being in a group of multiple kids. Most importantly for what I do, I'm in a department of psychiatry. Disorders are rarely diagnosed in that period. So it's rare that a child gets an actual diagnosis of something. But symptoms and temperamental variation are apparent. So, okay, let's talk about what I mean by that. So if you think about a child who's maybe middle childhood or adolescence that goes into a psychiatrist or psychologist for a diagnosis, they often ask the parent, okay, so how long has this been going on? And the parent will usually say something along the lines of well, for example, anxiety, it's really been, the last 6 months have been really bad, meaning it's become very impairing in the last 6 months. But they've always kind of been an anxious kid. This didn't come out of nowhere. And that's what I mean by that temperamental variation that we can study early on in development. But of course we have challenges, right? So children this age have limited verbal and cognitive ability. So anyone who's tried to have a conversation with a 4-year-old about their emotions knows it doesn't go that far, right? So we have to be creative in how we test these children and how we get information from these children. Also, there is not a clear line between typical and atypical behaviors. So think of something like tantrums, which I have studied in my work in irritability. Well, kids have tantrums. That's kind of part of preschool age development. But when is it a problem and when is it not a problem? There's kind of no clear boundary. So that's one of the things I study. Okay, so we're going to go through 3 parts of this talk that kind of takes you through all my career stages, actually. So first, we're going to talk about my early work and look at a developmental model of temperament to psychopathology transition. And I'll use my work in irritability as an example. Then we're going to talk about my more current work in the parent-child relationship and interpersonal neural synchronization, which is looking at how 2 brains synchronize. And finally, the work that I'm doing, like right now, like unpublished data, we just pulled to put in this talk, and look at the role of family systems and life stress on numbers 1 and 2. Okay. So let's start with the developmental model of psychopathology transition. So in order to understand the perspective that I take, I want you to consider 2 preschool children. The first is this happy child. We know him, we love him, he walks around all day looking happy like that. He has, the literature tells us, adaptive social interaction, positive home interactions, higher likelihood for school success. But then there's his counterpart, her, the irritable child, walks around all day, looking like that. She is more at risk for social problems, challenges in the home environment, higher likelihood of school failure, but most importantly from what I do, she's at about a 50% risk for the development of psychopathology when she is older. The problem is we don't know which 50% are which. Some will just be irritable adults, and some will go on to psychopathology. So the questions that we ask in my research are, what neural circuitry distinguishes these 2 children? Can we identify early neural dysfunction that predicts conversion of psychopathology for this irritable child, but maybe not this irritable child who goes on to develop normally? So let's think of her as the girl in yellow, as the girl developing psychopathology, the girl who develops normally is in red. So how can we use our knowledge of biological systems to intervene early enough to redirect the course of atypical development? So if we can start early, we can maybe prevent psychopathology before its onset. Okay, so here's the working developmental model that we use in my lab. So this is the normative case, the child who's irritable and goes on to develop normally. We would expect that across the age of 3 to 6, we would see a decline in frustration and negative affect. Those are the 2 parts of irritability. And we won't get too into them, but we expect normative development is that this declines over the 3 to 6 age range. This comes, for the developmentalists in the room, with that steep slope of executive function growth that happens between 3 and 6. So what we hypothesize is happening here is that executive function growth kind of is what lowers that irritable functioning and helps the kid regulate to leave a high likelihood of healthy outcome in the normative case. However, in the clinical case, what happens is that frustration and negative affect does not decline. So while red shirt girl, normative girl, might be still the most irritable child in her class, at the age of 6, she's the most irritable normative 6-year-old. Here, in the clinical case, the child at 6 is still acting like an irritable 3-year-old. What we think is happening is that the executive function is not developing at that steep slope that it should be, and that's not leaving a high likelihood for healthy outcome, not helping that decline of irritable symptoms. So I'm going to show you some of my work, just a little bit, that supports these hypotheses. What I'm going to show you mostly here is the method functional near-infrared spectroscopy. Do you guys, anyone here, use near-infrared? Okay, well, then I will give you an introduction. So it is it is measuring the bold signal, like fMRI, same thing, it's measuring blood flow, but it's doing it with near-infrared light instead of magnetic resonance. So in my lab we do both, but I'm going to show you mostly NIRS work today. So what this does is it's the light there is going through a little cap. So one of those, there's those sources and detectors. The source sends in the light to the brain The detector measures the light coming out of the brain using the Beer-Lambert law from physics. That tells us how much hemoglobin concentration there is in that particular region. It is a region of interest only method, so you have to choose your region and its outer cortex. It is safe, painless, can be used in newborn infants, less light exposure than walking outside on a sunny day. So the advantages over MRI are motion sensitivity. I could have my NIRS cap on right now, acting like this, and you could be, getting blood flow from my brain, whereas in MRI, we can have no movement. Location, I can bring it anywhere I want. I have 2 of them in my lab. I've brought it to children's museums, I've brought it to schools, it's very mobile, and it's more cost effective. They are expensive pieces of equipment, but once you own them and you can do whatever you want with them. It's not like an MRI where you're going to pay a per hour use. And they're interactive, so I'm gonna show you later how we can split the uptones on 2 different brains and look at how brains interact with each other. Okay. So I'm going to show you one study supporting this model. Before I show you the task, we design all our tasks in-house. They're all available on our website. You can look at many executive function, emotion tasks. We have probably, 10 to 15 at this point. I don't know if they're all online. But if you're interested in any of them, we share them freely with the scientific community. You can email me and I can get you set up to use these tasks. So this is we're looking here at irritability and one domain of executive function, cognitive flexibility, and this is the pet store Stroop task. So we, again, are using little kids, so we got to deal with the challenges of little kids, so we've got to make it fun and exciting. Also, little kids can't read, so they can't do a traditional Stroop tasks with letters and colors. So we designed the pet store Stroop. The kids are showed a little animation, told a little story about how they're a worker in a pet shop, and all the animals have escaped in their cages. And what the children have to do is get the animals back into their cages. So first, we have non-Stroop kind of baseline trials where an animal appears in the center, and that's a cat. Can you hear that? Yeah, you guys hear that? Okay, so it's a cat. It says meow. The child would have to touch the bottom corner, the cat tree, put the cat in the cat tree. Okay, but sometimes those animals are really tricky and they're trying to trick you so that you don't catch them. And so in the Stroop trials, what happens is the cat appears, but the cat says, okay, so careful everyone, it's not a cat, it's a dog. So that's a dog pretending to be a cat to trick you. So the child would have to put the dog in the doghouse. Everyone get why this is a Stroop? Okay, great. So, and we have many tasks just like this. Okay, so let's take a look at the data. This was back in 2016, so this is, older data. What we did was we had 46, this was early days of NIRS where we could have smaller samples. We had 46 children. I want to make sure it's clear, these are typically developing children. These are just recruited from the community. None of them have any disorders or symptoms diagnosed yet. So what we found here on the x-axis, their level of irritability in a normative sample, the higher the irritability, the more they used the dorsolateral prefrontal cortex during those Stroop trials. So the higher the irritable children, normative irritable children, the more they used their executive function. So what this is telling us in early support of the model is that highly irritable children who are functioning well have good executive function. They're using their neural resources. But that's interesting and everything, and I am a neuroscientist, so I love the brain stuff. But for me, it's also really important to apply this work out to the community. Like, okay, so what? Like, what do we? Dorsolateral prefrontal cortex activates. What are we going to do with that information? So what we did, and this, even though it has no brain in it, is I think my favorite study I've ever done. because of that translation to the community. So what we did is we went to the Pittsburgh Youth Study. You guys probably, Sarah might be familiar with it. Yeah, Sarah is, but I don't know if the rest of you are, so I'll give you a little bit of background. In 1987, 503 first graders were brought into the Pittsburgh Youth Study. There's been many, many waves. It is now thousands of kids, but this is the first wave. 61% were receiving public financial assistance. This was a study originally designed to look at juvenile delinquency in the inner city of Pittsburgh, which, by the way, I was faculty at Pittsburgh for 12 years. So we had easy access to this data when I was doing this. I was at Pittsburgh. Those, by the way, also, fun note, in 1987, I was also in first grade. So those of you who want to do the math on how old I am can figure that out. So these children who continue to be followed to this day are my age. They were assessed annually through young adulthood, and this is still ongoing. They were assessed every spring until age 14 by teachers. 8 time points. Want to make it clear? These are 8 different teachers, each year a different teacher at school, which speaks to the continuity of these constructs. So at age 16, they were brought into the laboratory to do a cognitive flexibility task. It's the Wisconsin card sort. And at age 28, we got a latent variable we're calling adult delinquency, which includes self-report of delinquent behaviors, diagnostic interviews, and criminal records. Okay, so what we've got here is a latent growth curve analysis where we found that there were 5 trajectories of irritability from the ages of 7 to 16. Okay, so we've got this high group, basically we're calling it persistent irritability. This red group, they're high, they stay high. The slope here is not significant, it's not going down. And we have the low group, they're low, they stay low, that's 57% of the sample. We've got some that are high and drop, we've got some that kind of stay in the middle, but we're really mostly interested in this low group versus this high group. What we did was, this is dimensional, but I'm showing this to you as a median split to visualize the data. What we did was we looked at their cognitive flexibility and delinquency outcomes. So let's take a look at the yellow first. So this is the low group. They're low in irritability, they stay low. So kind of typically developing. It doesn't really matter whether they developed good cognitive flexibility at the age of 16, they had the same delinquency outcomes. And keep in mind, this is an inner city sample. They are at very high risk for delinquency. However, look at the red group, the red group, if they were high in cognitive flexibility at the age of 16, they had the same delinquency outcomes as a low group. But if they had low cognitive flexibility, poor executive function, they had much higher delinquency outcomes. So this is, again, support from the community of this model, and I just love this study so much. Okay, I'm going to skip the MRI data, but if we have time, we can come back if anyone's interested, or if anyone wants to see it after this. I hear we have a little extra time in the room. So I'm going to skip the MRI data. So that we can move through the talk quicker. So let's move to number 2, examining the effects of the parent-child relationship. So what happened was, I was doing that research. We published a lot of papers, like that Stroop paper, in all areas of cognitive flexibility. And then I started to kind of say, or areas of executive function. I started to kind of say to myself, okay, well, yeah, we're looking at the child and temperament, and that's all interesting, but like, what's the next step? Kids don't develop, in a bubble. They have families, they have parents, they have neighborhoods, they have schools, they have all these things that influence how that executive function is developed. So what I started to do was think, let's take that model and let's put it into the context of the parent and child, in early life. But then also, we can look at the community context after that. And those of you developmentals are probably thinking, well, yeah, Susan, you didn't invent this. This would be ecological systems theory, right? So we have the child, that has a mesosystem, microsystem, exosystem, all the way out to these other systems, creating proximal influences on the child's development to distal influences on the child's development. So you're right, I did not invent this, but I've kind of seen my career as expanding from just that proximal child influences out towards the distal layers. So let's look at that next layer, the parent-child relationship. Okay, so why is this so important? Well, the child's primary attachment relationship is the parent, and attachment is our survival mechanism as a species. We don't fight or flight when we're born, right? Our mechanism for survival is attachment. Someone's going to take care of us. This can serve as a potential buffer against other stressors. We know that from the literature. Most importantly, in the world of psychiatry, at a young age, preschool age, any evidence-based intervention, mostly PCIT, parent-child interaction therapy is the most popular one, that consists primarily of parent training. So it's training the parent to interact with their child for better behavior. At the very least, a parent would need to be involved in their child's therapeutic relationship. So I look at a construct called dyadic synchrony, which again, I didn't invent. It is a mutually responsive and interconnected interaction style, and is linked to positive outcomes in the child development literature. So dyadic synchrony is that give and take of social interaction. So one person's talking, the other person's listening, eye contact, gesture. In early development, we see it very, very early, looking at how parents and children, the parent makes a face, the baby smiles. It's been used in the still face paradigm when the parent stops interacting and the baby loses it because they aren't getting that contingent responding. So this is something that human interaction is built upon. It's a tool for social communication and hypothesized to underlie attachment and bond formation. Okay, so what I did was I took that construct and thought, okay, well, if this sounds kind of crazy, but if two people have that behavioral contingent responding, their brains must be interacting as well. Their brains must have contingent responding too. So we're asking ourselves, is there an underlying neural component to parent-child dyadic synchrony? We're measuring the correlation between signals of brain activation of two interacting individuals. These can be any interacting individuals. What we can do is look at the real-time influence of parent behavior on the child brain. This can help us with therapeutic interventions. So what we can do is we can say, oh, look, when the parent does this, we're getting more dyadic synchrony. So maybe we design, therapeutic interventions around moments like that. And the NIRS equipment is great for testing this possibility, because what we can do is, this is one NIRS system, and what we're doing is we're taking these green fibers and we're splitting it between two brains. The system doesn't know it's on two different heads, so it decreases any measurement error from having two different systems. So NIRS is very, very good for this. Any other method has doing dyadic synchrony with other methods. It's been done with EEG. It's been done with other methods. It's harder to do because they have EEG has individual grounds. There's all sorts of difficulties there. Okay, so when we first started doing this with us and some other groups, we were like, this sounds kind of crazy, right? Like, do brains interact? So let's just do like a proof of concept study to see whether this is real. So what we did was we designed this task that's now used in all sorts of laboratories, and we use it in all of our studies. So what we did was we brought adults into the lab, two people who knew each other in some capacity, and we said, you're gonna do tangrams, these ancient Chinese puzzles. And so what we did was first they counterbalanced, they did the together condition. They're given a card of a, cat, whatever that is, and they're given 2 minutes to work together with 14 pieces on the table We just said, work together, do whatever you want to do, eye contact, gesture, whatever, talk. You have 2 minutes to get a certain amount of puzzles done. And it is hard. Very rarely can they get one puzzle done in this amount of time. Okay, so work together. Then we had an apart condition. What the apart condition did was it put the same 14 pieces on the table, so we're seeing the same stimuli, except the puzzle is now a 7-piece puzzle. So there's still 14 pieces on the table, but they're each taking 7 pieces. They're doing this rabbit. We say you're not competing, but just work on your own. Do the puzzle on your own. Same timing, same sound, same card, table looks the same, but the only difference, the manipulation is you're not working together anymore. You're not cooperating. Okay, we also, just to control for the sights and sounds of the room, we had a movie condition where they looked up, they saw on a screen 2 hands doing a together puzzle that would look like them looking down at their own hands with their partner, just to control for like seeing the same stuff. Okay, so what did we find? So what we found was, what I'm showing you is each one of these is a brain. This is the nose, this little triangle. When they're looking at each other, they're cooperating. When they're looking away, they're not. What we found was that we only saw interpersonal neural synchronization. Neural synchrony is another term people use in the together condition. So these lines are representing increased synchronization between the optodes. So this optode to this optode has that synchrony. Okay, but what we did to take it even a step further is permutation testing of non-concurrent pairs. So what we do there is we take a random person and we pair them up with every other subject in the study. And if neural synchrony is real, we should have increased neural synchronization only with their partner and not every other person who came in and did the same task and heard the same sounds and saw the same stimuli. And that's exactly what we found. You can see this, the together active condition, you can see this green bar here has increased synchronization. And now we've incorporated this permutation testing into all of our studies, just as a kind of a reality check. Let's make sure we're seeing real synchrony. So we have real dyads and false dyads, we call them. And that's like our first test that we always do. And we've never not found it, by the way. Every single time we find the permutation test to be significant. Okay, so let's go back to irritability. This was the first study we did in parents and children. What we did was we took those tangram tasks and we added we made it a bit of a mild stressor. So we modeled this after the disruptive behavior diagnostic observation schedule, which my colleague Lori Walkschlag designed, which is a test of disruptive behavior in the laboratory. So what we did was we introduced this task, we made it a little more stressful. We said, you only have this amount of time to do 5 puzzles, and most families are able to do this. And if you don't get a certain amount done, you won't earn a prize, and there's a countdown clock. And there's a nice set of really, really fun toys sitting right next to the child, and we say to the child and parent, oh, you guys can't play with those, you can't touch those until you're done with the puzzles. So, little kid, this is not fun. This is a bit stressful. It's a mild stressor. The parent's got to keep the kid on task. The kid doesn't want to do it. They want to touch those fun toys they've never seen before. So they do that. Sometimes we have a baseline condition before that too. And then afterwards we have the recovery condition. So after they've been stressed, we have the recovery condition where we time just like the stress condition, but they get to play with those toys in an interactive way. Okay, so it ends positively. All right, so what did we find in a sample of typically developing children? So what we found was that there was interpersonal neural synchronization in the negative interaction and in recovery, right? There should be. You're interacting in both. But that there was no relationship between neural synchrony and irritability during stress. So it didn't really matter how irritable the child was. everyone kind of got stressed equally, and there was the same amount of interpersonal neural synchronization. However, irritability negatively correlated with neural synchrony during recovery. So what happened here is that the higher the child, and this is controlling for parent irritability, the higher the child was in irritability, the lower their neural synchrony during recovery. What we think is happening there is it's taking longer for those irritable kids to kind of snap out of the stressor. They're not kind of, oh great mom, it's fun, let's go play, that's done, let's go play with the toys. They're kind of holding in that stress and that anger and not quite recovering in the way that the low irritability children do. Also, what we found is that neural and second by second coded, every video coded second by second of dyadic synchrony positively correlated during the stressful condition. So we got the brain and the behavior correlating during stress but not during recovery. So this is something we're talking about a lot now. When do brain and behavior overlap and when don't they? Sometimes they do, sometimes they don't. But keep that in mind, because at the end I'm going to talk to you a little bit about behavioral coding and what we're doing with behavioral coding, because it is so hard. Okay, so let's move on to part 3. And this is kind of where my lab is right now. Now we're looking at the role of family system and life stress. So we're going out again into that ecological systems theory, out into the later systems, and we are looking at the family and community context. So what we did here was we took that same larger sample, the one we were just looking at, and we took every questionnaire and interview that we had, and we did a factor analysis. Every questionnaire and interview we had on adversity, any type of adversity these families are facing, and we did this factor analysis, and we found that it loaded onto 2 types of adversity. The first one, which we're calling socio-demographic diversity, included the area deprivation index, which is, do you guys use that, something like that here? Okay, it's how your zip code is how much deprivation your zip code is considered to have compared to zip codes around you. Family income, social services utilization, single parent status, and caregiver education. Factor 2, we're calling familial risk. That included caregiver psychopathology, family conflict, and household chaos. What didn't load onto either of those is teen motherhood, because we just didn't have that many teen mothers in the study. I think we had like 3 or 4. So what did we find? Well, what we found during the frustration condition, during the stress condition, is that the higher the adversity in socio-demographic adversity only, the lower the neural synchronization during stress. So families experiencing high socio-demographic risk are having low neural synchrony during stress. But during recovery, it's equal. So that's great news, right? They're recovering normally. What we think that could be is there's many potential explanations. Some of them include like allostatic load. They're under a lot of stress. When more stress is added, they need to disengage a little bit. So, it could be a protective mechanism. But what's really important is that 5 minutes later, right back to synchrony. So this is important to consider when we look at therapeutic interventions. Okay. Now that's all early work. Now I want to move on to a study that just like last month ended in my lab. The first wave of it just ended. So this was a child affect and resilience to experiences study that we're calling CARE. This was a study funded by the government for 5 years of following children who were experiencing specifically familial stress. We recruited 4 to 7-year-old children, followed at 6-month intervals. We are examining the biological effects of parent conflict as an attachment-related stressor. So specifically, we're getting all stressors here, but we are specifically but we're specifically looking at parent conflict. So what we did was we recruited families who in the last year the parents had either gone through a divorce or had had an increase in parental disagreement conflict in the last year specifically. So not just divorced families ever. In the last year, so this is a recent stressor. And then we had control families who are experiencing low conflict and or never been divorced. So what we measured in this study, a lot of data, neural stress circuitry using fMRI and DSI, neural synchrony using NIRS, cortisol, hair and saliva, and psychopathology outcomes. We also measured all other environmental stressors because those do come along with parental conflict. So let's take a look at the sample. This is all very new, unpublished data. So this is, one of the first times I'm presenting a lot of it. So this is 231 kids. We have the high conflict group and the low conflict group. High conflict is either divorced in the last year or increase in conflict in the last year. Low is opposite. So it's about 50-50. So what we have is that they are equal in demographics. They're equal in race and Hispanic ethnicity. They are equal in socioeconomic status. But interestingly, when you use the SES community ladder, I don't know if you guys use it, which is a measure of how you feel about your SES. So how you perceive yourself. It's asking how do you feel relative to the people around you. The conflict group rates themselves as having lower socioeconomic status, even though they don't. They are higher on parental conflict. They are also higher on ACEs, not including the divorce item. So the ACEs has, 12, 16, I can't remember, items of life stressors. If you remove the divorce item, the high conflict group still has 2.2, it's really more like 3.2 ACEs. That's a lot when you're like 5 years old. You haven't had a lifetime to build up all these stressors. So this is a very high stress group. And the parents are reporting higher stress on the perceived stress scale, which is expected. So taking a look at the parents versus the child, the children are higher in functional impairment. They are higher in internalizing symptoms, higher in conduct and ODD, and higher in ADHD. The parent is reporting lower instrumental support, lower emotional support, higher depression, higher PTSD, and higher anxiety. So this is a high stress group here. Okay, taking a look at some early cortisol data, we looked at the cortisol awakening response. That's salivary cortisol when you first wake up in the morning. A recent divorce with higher parental conflict, so the conflict being high between the parents even after the divorce, was associated with a higher cortisol awakening response. And there was also that increase in cortisol awakening response over time, over those follow-up compared to the controls. So they're experiencing higher cortisol. I'm gonna skip the MRI, but we can come back if we have time. Let's talk about dyadic synchrony as a buffer. So the original hypotheses for this study was that for families living in stress, specifically parental conflict, a supportive parent-child relationship will buffer against the development of psychopathology symptoms. So we're looking at that parent support as a moderator, a buffer. We are looking at dyadic synchrony as a tool for effective social interaction and bond formation between the parent and child, and how that comes as a buffer. And we are looking in this early data at 3 variables in our study. Stressful life events, that would be the ACEs, perceived stress, that's parent-reported perceived stress, and parental conflict. Okay, So what we did was we had that same task that I showed you with the puzzles. We have a baseline, we have a stress condition, and we have recovery. We've plotted every single one of these families. Okay, the 231 families. So this is how they all looked. Then what we did was a latent profile analysis where we found that 162 dyads fell into this blue normative profile. That's their average. The black line, by the way, is the group average. And we had 36 families fall into the low synchrony profile. That's lower than normal synchrony. That's this orange right here. So what we did was we then looked at the ACEs, the perceived stress, and the parent conflict. Parent conflict didn't predict anything. So what we're looking at is, I'm sorry, I'm new. This is the first time I'm ever showing this. So this was like last week we finished this. So I may not explain it that well. But what we found was an interaction in predicting internalizing, externalizing, and impairment. None of this came out for parental conflict. It was, however, for the ACEs and the parental stress. What we found, these are our significant interactions. When you are part of the normative profile, it doesn't your synchrony, when you're, okay, let me explain this better. When you are low in synchrony, you are more likely to have externalizing impairment impairment and externalizing if you have high perceived stress and ACEs. So being low in synchrony puts you more at risk for externalizing impairment and impairment in both categories. Did I explain that well? Sorry, it's my first time ever explaining it. It's still pilot data, so we're still working with it. So this is kind of some evidence of synchrony being in the normative profile of synchrony, serving as a buffer against these poor outcomes. Okay. All right. So let's talk about some new directions with this work and other things that are going on in my lab. First, the care renewal. So we're taking that sample and we're applying for funding to keep following them. The care renewal is under review with a focus on family conflict, predicting anxiety, depression, and suicidal ideation as these kids transition into middle childhood. So we're moving to all of family conflict, not just parental conflict. We're considering the children more active at the middle childhood age that they've now aged into. And we're looking at the specific mood and anxiety outcomes. We are including epigenetic aging in the care sample, which I'm working with Alicia Smith at Emory, who's an expert in this, to look at how their epigenetics in their body actually age as a function of these early stressors. We also have another RO1, which is a longitudinal study in a big NIH-funded study in my lab, which I didn't talk about today, that's looking at the transmission of anxiety through dyadic synchrony. So we maintain in my lab that dyadic synchrony is a tool. It's a tool for social interaction. It isn't good, it isn't bad. It should come and go. It's dynamic. And we are looking at the potential of there being moments where it's not adaptive, it's not helpful. So what about situations where an anxious parent is in a stressful situation with a child? Do they transmit that anxiety through dyadic synchrony? And we are looking at this because anxiety is only moderately heritable genetically, yet the correlation between parent and child anxiety is fairly high. So there have to be behavioral pathways whereby anxiety is transmitted from parent to child. So this is ongoing in my lab. We're also doing some early research in pain in early psychopathology, specifically physical pain, as an indicator of early stress on the body before psychopathology onset, because children often report things like headaches and stomach aches before they report psychopathology. We are also, this is the last thing I'm going to show you, developing AI models for dyadic synchrony coding and brain integration. So back in the day, 10 years ago, the data I showed you, we had to code second by second the parent-child interaction, and that took hours and hours of labor. And, there's also, I don't know if any of you here do behavioral coding, but it's just so hard. It just requires such a workforce, and, there's coder drift and all these problems. So what we are designing here is an AI model. So this is where we're at so far. So the AI model will code dyadic synchrony for us using our dyadic synchrony coding system at that second by second level, and then integrate it with the neural data. So we have this package we're creating. It is not available yet. We're working on it. We have some funding from WashU to do this. It's called Synchron AI. Like synchrony. My staff thought of that. I thought it was cute. And they will be a package where you can use behavioral, you can use neural, you can integrate them, and it will be publicly available. So what we have so far, this is in only about 40-something coded videos. So very early. We're going to be spending the next year coding more videos and training them out even more. We have about a 75% accuracy rate in predicting dyadic synchrony, meaning predicting the coder's dyadic synchrony at each second. We have about a 75% accuracy rate. We have these heat maps for exploring useful spatial features. So what this is saying, so this is second by second through the video of the parent-child interacting. And what this is showing us is what the AI model found most relevant to making to its codes. So what it is saying is, like, this screen in the back, not useful to me. That's not helping me with synchrony. Door, maybe slightly useful because a person does walk in and out to change the toys, and they both look joint attention. So you get a little bit here, particularly you'll get some when someone comes in. But what it's really saying is what's useful to me, if we're going to personify the AI, what's useful to me is this table where we're moving pieces around or toys around, where their hands. And that's because joint attention. They're in this particular test, they don't look at each other's faces a lot. They're sitting next to each other, and they look at each other's hands when they talk. But what's this here? That's the cart of toys. So you would think, okay, well, why? It doesn't move. They're not allowed to touch it. Because it's an element of joint attention. The kid looks over and says, oh, I want to play with that, Mom. Mom looks over and says, no, we can't. We got it. We have to finish this first. They're both attending to the same object, which is why this is useful to the AI, even though it doesn't move and doesn't do anything. So, we're leveraging some pre-trained models. And I would like to thank you for listening to my talk. Thank you so much. And yeah, I'm happy to answer questions. These are the members of my lab and collaborators who have been helpful in this research program. We thank the children and families who participate in our research. This is the funding of all the studies that I showed you. And yes, I thank my group, and thank you so much for having me. Yeah. So I can take questions. I don't know how much time we have. Yeah.
Yeah, I think we have at least 5 minutes, but probably 10 minutes for questions. And please, the ECRs in the room, please do ask some questions.
Yes,
as well.
yes, I want to hear from students for sure. Any questions? Yeah.
Hi, I'm curious. With those synchrony experiments, was it typically mother that was the parent, or like other caregiver? Did you find any differences?
Really interesting question. So the PCAT one, the anxiety transmission study, has 40% of the sample is fathers. So it's still ongoing. We don't have the data on that yet. But personally, I've never, like, in all my years of doing this research, I have never found a sex difference, like, ever in anything. Like, synchrony is a basic human interactive construct. I don't think we're going to find a whole lot of difference between mothers and fathers, but it is one of the questions of the study. In the work that I showed you, it was whichever parent chose to participate. It was mostly mothers, but there was no rule on who could and could not participate. Yeah.
Just to kind of add to that,
Yeah.
because I think some of the work that maybe you might be familiar with is synchrony when people are doing a not joint attention task. So there's work that shows, like,
Yes.
relationship closeness when you're doing something like watching a movie.
Yeah.
Can sync neurally, but this is because they're doing the thing together.
Yes.
It's different.
So that is a different, so what's challenging in this field is that people use the term synchrony to describe a lot of different things that are not the same thing. I was about to show you, let's see if we go back here. This work in MRI, I won't go through it, but this is called intersubject brain synchrony. It's a type of analysis that's being done. It's not at all what I'm talking about. I didn't make this term, but it's a totally different thing. It's exactly what Sarah's talking about, how one person's brain is like another person's brain when they're watching the same thing. Yeah, so spoiler, it is lower in the children from families with conflict. Yeah.
Really interesting talks.
Thank you.
I'm wondering about the synchrony coding, I suppose.
Uh-huh.
Is it truly synchronous, or is it somebody responding quickly to someone else? And then if so, what direction? So we know that parent-child interactions are bi-directional.
Uh-huh.
It's not just the parent dictating how the child is always responding. And so does the child, is the child sometimes responding to the parent?
Yeah, so the way that we code is we code second by second, and you have to have 3 seconds of what, and there's a whole, like, manual of what synchronous engagement is. You have to have 3 seconds of synchronous interaction before you start coding a time period of synchrony. So it goes in and out. Every dyad, no one's in 100% synchrony. It's going to go in and out for everybody. Our coding system at the moment does not code who a lead or a follow. Because synchrony takes 2. You're either in synchrony or out. So it doesn't currently, like the human coding that we do, which is time intensive enough, is not getting that. We have discussed, once we get the AI up and running, that's something we could code, because we're looking at using the AI models of behavior and language. That would be something much easier for an AI to do than for a person to do. So we hope to have that as part of the AI package. Yeah.
Yeah, I was just thinking in terms of using that to guide the interventions.
Yes.
Really intervening on the parent, but what about the child's ability to respond to social cues?
Yeah. So what we can do once we have this AI work up and running, what we can do is we can have it find specific moments of the interaction where either a parent does something, like an example would be like parent touches child's shoulder, or a child does something, and how does that affect the synchrony. So we can do that with the AI models, but it is just, at the human level, we have not been able to do it. Just the second by second dynamic, time course is just so hard to deal with without high-level tools. Yeah. Yes.
Great.
Thank you.
I had a question about the expert study.
Yeah.
What sort of behaviors were being considered as delinquency, and like how is that being measured and who's reporting?
Yeah, so that was a latent variable. So it included both questionnaire reports of their delinquency and psychopathology. So it included psychopathology also. A clinical interview. Probably the SCID, I can't remember, but probably the SCID. When they were younger, probably the case ads, because that was also developed at the University of Pittsburgh. So the case ads and the SCID went at youth, and then older, the SCID. And their criminal records were also pulled. So that was a latent variable including all of those. Yeah.
Thanks for a great talk.
Thank you.
In the earlier work, you were talking about executive function
Uh-huh.
and named irritability.
Mhm.
In the later one, are you still looking at that? And does it play a role like in the synchrony, for example, or like kind of the resisting the negative effect of adversity?
Yeah, so actually I didn't show the data. In the PCAT study, the parent-to-child anxiety transmission, I don't think it's out yet. I think it's been accepted for publication but isn't published yet. We have a paper coming out that is showing increased synchrony with increased executive function in that sample. So yeah, we are still looking at executive function. I'm not doing as much irritability work these days. I'm still interested in it. I'm still like one of the chairs of a conference a small conference on irritability that we do every couple of years. But I kind of got to a point where I felt like, okay, I've done this. I'm still interested. I still collect the data, but I'm no longer applying for grants in irritability only. I've more moved out into this this parent-child environment work. Yeah. Yeah.
I have a question.
Oh, yes.
Did somebody else have a question? Go ahead, no, please, you exactly.
But someone asked a question about gender difference in
Uh-huh.
the synchrony study.
Uh-huh.
I was wondering if you ever considered putting two parents at the same time, if that's possible with
It is possible. We haven't done it. We could do it. What it does, though, is our NIRS system only has a certain amount of optodes. So the trade-off would be you can measure, if we used just the system that we have, we would have to now go down by a third to the regions that we can measure. And then it makes it much harder to measure, you get the analytic issues of, child to one parent, child to two parents to each other. So yes, it's doable. We haven't done it, but maybe someday. Yeah. Yeah.
Hi, I don't know this time, but I'd love to see
No,
the MRI results.
Oh, okay. You guys, I know the room is available. I'm happy to do it.
Yeah, why don't, if anybody needs to leave, feel free, and then maybe we can chat about that. I was just going to piggyback on that.
Yeah.
Because people are getting up. Perhaps you could use parent-parent synchrony as a measure of conflict.
Yeah,
So you
yeah.
could do a sort of thing where they have to do the puzzle together as an in-lab measure of how well they communicate.
We've talked about that. One of my close friends, Brooke Feeney at Carnegie Mellon University, studies attachment in adult relationships.
Yeah.
So she studies, she follows couples to, see basically who's going to break up. And we have talked about doing things like that. We haven't quite incorporated the parents and the kid yet, but yes, this is something that we've discussed. Yeah.
Okay, I feel like the disruption has abated, so if you want to show some of the imaging results.
We have one more question, and I could show images.
great presentation.
Thank you.
But you still have the question about gender and question as well. Have you considered different sex pairs, mother, daughter, son?
We have the data. Any time I've run, mostly up until the study that's ongoing where we've recruited 40% fathers, it's mostly been mothers, because that's just who brings our kid in. Not exclusively, maybe 10% fathers, but mostly. Anytime I've looked at a daughter versus son in anything I do, irritability work, anything, I find nothing. I find no sex differences. and I don't expect to really. I don't really have hypotheses that, you know, this is, it's such a basic social construct. I don't know why it would be different in mothers and daughters, or mothers and sons, or what. And remember that synchrony ebbs and flows. Synchrony isn't like a personality. It's not, and some labs disagree with me, we can get into that, but it's not like these people have a, you know, 80 synchrony score. That's their, it's a tool. You go in and out of it. It's not a personality. So I don't know how we would expect a difference between the child sex. I also don't really expect a difference between parent sex, honestly, but it was something the reviewers really liked, so we added it to the grant. And it's in collaboration with Darby Saxby, who's like an expert in the father-child relationship. Yeah.
Just to follow up on that, I wonder whether the differences might not be necessarily in the neurosynchrony, but whether or how much synchrony there might be, but the type of synchrony. So I can expect that some of them might be more language-based versus action, shared action, or shared looking in terms of the different genders. And I wonder whether that
could be. The way we've coded so far, well, neural synchrony is just neural. The way we've coded dyadic synchrony so far, it's a conglomerate. It's what the coder thinks that second. Are they in or out? It's yes or no. Are they in or out of synchrony? The AI model can do things like that. A human could never. So we'll see. That's a good question. We can look at that, something like that with the AI model. And it's going to vary by task. Like the one I showed you is much more movement based. There's not a huge amount. I mean, they talk. They don't make eye contact. They don't like. It's a task set up for a very certain type of synchrony. And then there's other tasks that we've done that are a very different type of synchrony. So it just depends on that. Okay. MRI, doing it? Okay. So let me so I was going to show you 2 MRI studies. So let's do this. Play from current slide. Okay. So this was after the community sample study. Okay, so what we did, this was still looking at executive function, irritability, interaction. What we did was we had a sample, this was a while ago, so we only had 36 children. We did a naturalistic emotion viewing study. So what we did was we very, very carefully curated video clips demonstrating different types of emotion. So we had positive clips, and these were all from children's videos. Positive clips, where there's song and dance and happiness. Negative clips, where there's, you know, fear and anger in the movie. And neutral, which was like more nature videos, like flowers blooming, dolphins swimming. These were very, very carefully curated. We coded every one of them for the amount of time on time a face is on the screen, the amount of song, the amount of people present in the interaction. So they're all very well matched to each other. Like the negative is very equally matched to the positive in everything except the emotional valence. So what we did was we took a sample of children 4 to 10 years. We took the child behavior questionnaire, with a measure of irritability. One of is a measure of temperament. We used the irritability subscale. And these children are typically developing no history of psychiatric diagnosis. What we found was an age by irritability interaction in the caudate middle frontal gyrus, which is a more of a like emotion processing but still executive function also region, and the superior frontal gyrus, which is an executive function region. What we found, again, this is dimensional, but I'm breaking this down for you guys in well, not median split, the top 75% of irritability in the bottom, the top quartile and the bottom 3 quartiles. So what we found was that low irritability, it didn't matter how old they were. They had the same activation in the negative compared to neutral. But the higher irritability kids, the older they were, the more they used these regions. to process negative emotion. And we think this is this is more support for these high irritability kids who are typically developing with no diagnosis, are using high levels of executive function to regulate emotion during negative clip viewing. And if anyone wants to use these clips, we encourage it because we spent so much time curating these videos. And then I can show you the last bit of MRI data is from the CARE study. This is more, okay, let's see, play from current slide. This is, again, unpublished data, very new. So what we did here was we, keep going? Okay. What we did here was we chose a children's movie series, a children's like TV series called the King Julian something. I don't know. It's on Netflix. The reason we chose it is because this lemur right here, he's a high emotional intensity lemur. This guy. You know this? Oh, okay. You're like the first person who's ever heard of this. This guy goes from like 0 to 100. Like this guy goes from like happy to afraid to, he has a lot of emotion. This is a high emotional intensity change series. So what we did was we had the kids lay in the scanner, watch an episode, just watch it, do nothing but watch it. And this is the kids in the CARE study. So this is the kids who are exposed to parental conflict. So then what we did was we had 10 coders watch the same, outside of the scanner, watch the same video using a joystick that we programmed. So their screen would look like this. They're watching the video, they've got a joystick in front of them, they would see this blue bar. They're told the entire time you should just be like moving the joystick while you're watching this kids series. When the emotion is positive, it goes up. When the emotion is negative, it goes down. When there's neutral, it's right in the middle. So you should just be like moving the joystick. So what we did then was we took the 10 coders, this is just 100 seconds just to demonstrate for you. We took each coder's time series. I mean, just visually, you could tell that they're very like in sync with each other. But what we did was we calculated the interclass correlation, downsampled to the TR of the scanner, which is 0.83 seconds. So we created 0.8 second time samples. We calculated the interclass correlation, which is at 0.71. So fairly good. It ranged from 0.4 to 0.85. The parts of disagreement were when an emotion changed quickly, because some coders, are a second before the other coders. But overall, it's very good agreement. You can see that black line represents the average time course. So what we did was we take the average time course, we regress it onto the time course of neural activity in all of our subjects, and we calculate something called intersubject brain synchrony. Keep in mind, this is not the same synchrony I was talking about previously. This is what I think is a poorly named analysis. I think it should be called intersubject brain similarity, would be a better term. What you're looking at is how much an individual subject's brain is like or similar to the rest of the group at each time point. So think of it as intersubject brain similarity, but you'll see it called that in the literature. Okay, so what we found, we're going to look at age, conflict group, and age by conflict interaction. So what we found is a significant effective age. The older the child, the more intersubject synchrony in the sample, which is what you expect. We found that in our first study too. Remember, I just showed you an age correlation. So as you get older, your processing of emotion is more like the other people of your age. We did not find a significance by conflict group. So the conflict group are, it doesn't matter whether you're in the high conflict group or the low conflict group, you're no more like each other. However, what we found was this age by conflict interaction in the dorsomedial prefrontal cortex. So the older you are, if you're in the low conflict group, we see that intersubject intersubject synchrony / similarity getting stronger here. But if you are in the high conflict group, you don't get more similar to your peers. You're still processing negative emotion, specifically negative emotion, in a disparate way. They don't process it like each other. Everyone's kind of variable. So this is, still all unpublished data, so we have a lot to do before we publish it, but that's the MRI results in the CARE study.
So how are you interpreting that? Is it to do in a developmental context, like the older you are, the more you might be regulating your emotions with your prefrontal cortex, which is more developed?
Yeah, well, so here, at this point, the data that I'm showing you is, I just want to make it clear, the data I'm showing you, this is cross-sectional. This is at the first time point. We do have the other time points. That's not here yet, so we will look at the change within subject. So yeah, we're looking at it. I mean, this was from like last week, so we're still talking about it. We're looking at it as you should be, as you're getting older and your prefrontal cortex develops more, you should be more similar to other people your age as you learn more about emotion and process it. But these children might not. They might be experiencing so much stress in their lives. And remember, it's not just parental conflict. They're experiencing a whole lot of other stressors that come with parental conflict. They're experiencing a lot more stressors and may not be developing in the same way. This is cross-sectional, so I don't want to make that conclusion until we've looked at it. I don't want to make a longitudinal conclusion on cross-sectional data. We do have the longitudinal data, but yeah, the fact that they're not becoming similar to each other or similar to their non-stressed peers could tell you a lot about how they're perceiving emotion in the world and how they're interacting with the rest of the world, and could tell you about why they're at a higher risk of developing psychopathology because of their emotion processing, or vice versa. And we will have all that data as we just finished data collection about a month ago, so.
It strikes me as a potentially like sensitivity, specificity question.
Could be.
So it's like they may be responding to the emotion change, but they may have less emotion differentiation, or they may be less nuanced in their emotional interpretation, which is likely driven by high conflict, high emotional responding, but not necessarily sensitive emotional responding. That is nuanced.
That's a good explanation too.
Yeah, so you would expect the higher synchrony is perhaps related to greater differentiation as they get older.
Yep. That's a good. I hadn't thought of that explanation, but that's also one.
Okay. Thank you so much, Susan.
I get to talk to you after.
I feel like please feel free to continue.
Yeah, absolutely.
After, but maybe we'll thank Susan first.
Yes, thank you so much. Yes, and I'm happy. I got nothing to do today, so I'm happy to discuss anything with anyone who wants to.